Good day, everyone. I am Kyle Angelica from the Bachelor of Secondary Education, major in Sciences. And today, we will be discussing under this topic which is entitled Meiosis. But before we will moving on through our further discussions for today, let's have first our learning objectives. So here's our learning objectives. At the end of this topic, you will be able to define the process of meiosis. Second, identify and describe the stages of meiosis. Third, explain the importance of genetic variation. And lastly, compare and contrast the differences between meiosis of males and females. So what is meiosis? Meiosis is a process where a single cell divides twice to produce four cells containing half the original amount of genetic information. So these cells are our sex cells. These are the sperm in males and eggs in females. So during meiosis, one cell divides twice to form four daughter cells. Take note that a cell is a basic structural, functional, biological unit of all living organisms. Cells provide structure for the body and take in nutrients from food and carry out the important day-to-day functions for life. So these four daughter cells only have half the number of chromosomes of the parent cell. So these are called dihaploid. So take note that a chromosomes is a thread-like structure in our cells and made along DNA molecule that wrap around a protein scaffold. So humans have 23 pairs of chromosomes. So other organisms also have chromosomes, and most bacterial chromosomes are loops or circles are DNA. So meiosis produces our sex cells or gametes. So these are the eggs in females and sperm in males. So when we say gametes, it is a male or female reproductive cell that contains half the genetic information for an organism. So in humans, the gametes are the sperm and egg cells. So meiosis can be divided into 9 stages, but this can also divided between the first time the cell divides, in which it is the meiosis 1, and the second time it divides, in which it is called meiosis 2. So here are the stages of meiosis. So, as you can see, in meiosis 1, it shows how separates homologous chromosomes. So, in interphase, as you can see on the structure, on the first side in which, or the first stage in which we called interphase, so the DNA in the cell is copied, resulting into two identical full sets of chromosomes. So outside of the nucleus are two centrosomes, in which each containing a pair of centrioles, and these structures are critical for the process of cell division. So when we say cell division, it is when a cell is splits into two daughter cells and each containing the same genetic material. So during interphase, microtubules extend from these centrosomes. So the next stage will be the prophase 1. The copied chromosomes condense into X-shaped structures that can be easily seen under a microscope. So each chromosome is composed of two sister chromatids containing identical genetic information. And the chromosomes pair up so that both copies of chromosome 1 are together, both copies of chromosomes 2 are still together, and so on. So The pairs of chromosomes may then exchange bits of DNA in a process called of recombination or crossing over. And at the end of the prophase 1, the membrane around the nucleus in the cell dissolves away, releasing the chromosomes. So the meiotic spindle, consisting of microtubules and other proteins, extends across the cell between the centrioles. So, the next stage will be the metaphase 1. The chromosome pairs line up next to each other along the center or the equator of the cell. The centrioles are now at opposite poles of the cell with the meiotic spindles extending from them. The meiotic spindle fibers attach to one chromosome of each pair. And the next stage of meiosis 1, in which we call the anaphase 1. So the pair of chromosomes are then pulled apart by the meiotic spindle, which pulls one chromosome to one pole of the cell and other chromosomes to the opposite pole. In meiosis 1, the sister chromatids stay together, and this is different what happens into mitosis and meiosis 2. The next one is the telophase and the cytokinesis. So the chromosomes complete their move to the opposite poles of the cell. So at each pole of the cell, a full set of chromosomes gather together. A membrane forms around each set of chromosomes to create new nuclei. The single cell then pinches in the middle to form two separate daughter cells, and each containing a full set of chromosomes within a nucleus, and this is known a process of cytokinesis, in which two haploid cells and chromosomes are still double. Now let us move to meiosis 2, in which it separates sister chromatids. So in prophase 2, as you can see on the illustration, now there are two daughter cells, each with 23 chromosomes or 23 pairs of chromatids. In each of the two daughter cells, the chromosomes condense again into visible X-shaped structures that can be easily seen under a microscope. So the membrane around the nucleus in each daughter cell dissolves away, releasing the chromosomes. So in this stage, the centrioles duplicate and the meiotic spindle forms again. In this, the next stage will be the metaphase 2. In each of the two daughter cells, the chromosomes or the pair of sister chromatids line up end to end along the equator of the cell, in which equator or the center. So the centrioles are now at the opposite poles in each the daughter cells. So meiotic spindles fibers at each pole of the cell attach to each of the sister chromatids. And the next stage will be the anaphase 2. The sister chromatids are then pulled to opposite poles due to the action of the meiotic spindle. The separated chromatids are now individual chromosomes. And the next telophase, the next stage will be the telophase 2 and the cytokinesis. The chromosomes complete their move to the opposite poles of the cell. So at each pole of the cell, a full set of chromosomes gather together, and a membrane forms around each set of chromosomes to create two new cell nuclei. So this is the last phase of meiosis. However, cell division is not complete without another round of cytokinesis. Once cytokinesis is complete, there are 4 granddaughter cells, and each with a half of the set of chromosomes, in which we called a haploid. So in males, these 4 cells are all sperm cells, and in females, one of the cells is an egg cell, while the other three of the polar bodies are called the small cells that do not develop into eggs. So this is how we connect or we gather the stage from the interphase up until the telophase, or down until the telophase 2 and the cytokinesis. So let's move on to the importance of genetic variation. Meiosis and genetic variation, in which meiosis is an important process that allows for the rapid generation of a new genetic combinations. So there are three mechanisms make key contributions to these genetic variations. These are the independent assortment, the crossing over, and the random fertilization. When we say independent assortment, it is the random distribution of homologous chromosomes during meiosis, which is called the independent assortment. So as you can see in the structure below, the possibility 1 and the possibility 2, in which the metaphase of meiosis 1. It shows the arrangement of chromosomes in each of the cells is equally probable. So in metaphase of meiosis 2, the gametes with different possibles, showing the gametes with different possible of combinations. The next mechanism will be the crossing over. So the DNA exchange that occurs during crossing over adds even more recombination to the independent assortment of chromosomes that occurs later in meiosis. Thus, the number of genetic combinations that can occur among gametes is practically unlimited. Furthermore, the zygote that forms a new individual is created by the random joining of the two gametes. The next one will be the random of fertilization. So, meiosis and the joining of gametes are essential to evolution. So, no genetic process generates variation more quickly. The pace of evolution is speed up by the genetic recombination, and the combination of genes from two organisms results in a third type but not identical to either parent. So by looking of the meiosis in males, it is a process by which sperm are produced in male animals, which is called the spermatogenesis. When we say spermatogenesis, it occurs in testes, in the male reproductive organs, and it produces gametes called the sperm. So during the sperm formation, the cytoplasm is divided equally after the first meiotic division. The cytoplasm is divided equally again after the second meiotic division. Thus, 4 sperm result from each cell that begins meiosis. So, meiosis in females, it is a process which gametes are produced in female animals, in which we called the oogenesis. So when we say oogenesis, it occurs in the ovaries in the female reproductive organs and produces female gametes called the ova. So during egg formation, the cytoplasm is divided into the unequally after both meiotic divisions. And 1 large cell and 3 smaller cells will be the results. And the three smaller cells die, thus only one egg or ovum results from each cell that begins meiosis. So as you can see, these are the difference between the meiosis in males rather, and female animals. So that's it for today. Just always, I will leave you a quote in which genes are like the story, and DNA is the language that the story is written in which it was quoted by Sam Kian. So that's it for today. Thank you and God bless.